సార్ మీట్ చేసిన ఎట్లా ఇన్వైట్ చేసిన okay chand. So good afternoon to everyone. So this is the 6th day so afternoon session by Professor K. Arun Kumar. So Department of Plant Science, School of Biological Science from Central University, Kerala. So before handovering the session to the Sir Professor K. Arun Kumar, so let me introduce the Professor K. Arun Kumar. So Professor K. Arun Kumar completed MSc and MPhil Botany from Bharati Darshan University and PhD from Center for Advanced Studies in Botany, University of Madras, India. After a short stint in Rubber Research Institute of India from 1997 to 1999. After that, Sir started his career in teaching botany in the year
botany
as assistant professor.
Botany.
Since 2007, working as so present position is Professor and Head, Department of Plant Science, School of Biological Sciences, Central University of Kerala. So Sir research interest has been in the field of diversity, biology and biochemistry of algae. So special focus is on isolation of biomedical compounds from algae biomass production and energy conversion, and carbon encrustation by calcareous algae. Sir is also interested and familiar in organic farming techniques, and also taught the subject biomass characterization in Swayam MOOC course. So Sir research experience is MPhil 15 awarded and PhD 4 students awarded and 6 students are under the supervision of Professor Arun Kumar sir, and research projects completed around 1 crore 15 lakhs, around 1 crore 16 lakhs. So sir is having the different member in the professional bodies and also active member in the academic activities. So chairman, Board of Studies, MSc Botany, Central University of Kerala. Convener, Anti-Discrimination Cell, Central University of Kerala. Member, Academic Council, Central University of Kerala. Member, Board of Study, MSc Plant Science, Central University of Kerala. Member, Board of Study, MSc Biochemistry and Molecular Biology, Central University of Kerala. Member, IQAC Core Committee, Central University, Kerala. Member, NAC Core Committee, Central University of Kerala. So, sir organized conferences 3 and written book chapters 5. So, research articles published in the last 3 years, total 52. So, we have a vast experience in the of biofuel production from algae. Sir, so over to you, sir.
Thank you. Thank you very much for the introduction given by the organizing secretary. Shall we start?
Yeah, sir, please.
Okay. So I am very much happy to.
എടി മലയാളി നീ വന്നേടി ചെത്തില്ല സുവോളജി മലയാളി ലൈറ്റ്
I am very happy to
ഐ എം വെരി
interact.
മച്ച് ഹാപ്പി ഐ എം വെരി ഹാപ്പി
To interact the participants with my background of research, and this opportunity, I think, since I am the purely a biology man, so I can able to influence the matter how the algae are or can support the biofuel production, considering so many factors. So it may be most of the participants here are uh, engineering background. Okay, but my presentation may give a biology information. What are the requirement? What are the role of this plant biomass, especially algal biomass, can support the sustainable fuel production? Uh, my slide is visible. Hello.
Sir, screen is sharing.
Sharing.
Yes, sir.
It is okay. Hello.
Sir, still sharing, sir. Still sharing,
Still
sir.
sharing, sir.
No, I am sharing. Whether it is visible.
Sir, no, sir, no, sir.
No, sir, it is not.
Now it is okay.
Sir, no, sir. Still screen sharing, sir.
I think, can you permit me to share that option? Have you given me option of sharing the slide?
It's already given, sir, but it is sharing maybe low bandwidth or internet slow.
Oh. Wait. Wait, it will be ready another 5 minutes.
సార్ సిద్ధార్థ్ సార్ హలో సార్
ఉపేంద్ర
సార్
Actually-
సార్ ఒకసారి ఏమన్నా లాగొట్టి మళ్ళీ ఒకసారి జాయిన్ అవ్వమని చెప్పండి సార్ ఏమన్నా
అదే ఏదో షేరింగ్ అని వస్తుంది కానీ స్క్రీన్ రావడం లేదు.
అదే సార్
Actually,
ఒకసారి జాయిన్ సార్ని ఒకసారి మళ్ళీ ఎగ్జిట్ అయ్యి మళ్ళీ జాయిన్ అవ్వమని చెప్పండి సార్
Exit ஆயிட்டு திரும்ப பண்ணுமா? Wait a minute. I will, please, sorry for the inconvenience. Wait a minute. Okay. Post participants. Actually, hello, it shows that I am disabled to share the screen.
No, sir. Permission is given.
Host disabled participant screen sharing. You make me as a host, then only I can share.
So co-host is given, sir. You can share now.
Now only it is coming.
Now it is okay, sir.
Now it is okay.
Yeah.
So earlier I was not given, I think that is the reason. I don't know. Okay. So now it is okay, no?
Okay, sir. Okay, sir.
I am very sorry. See, this is my brief information about my academic credential. I am mainly focusing on algal research, especially drug development from algae and biofuel production from algae, especially ethanol and biodiesel. So let me see today. I will divide my presentation into two parts in biofuel production from algae. So how the algae is a promising plant group among the plants to support the biofuel production. Then the next part, how algae can practically meet the demand of biofuel for future sustainable biofuel production. So I will divide the talk into two parts. First part, we will see what are the potential of algal biomass for biofuel production. As we all know that the temperature, ambient temperature keep on increasing. If you see 2100, there is a possibility of increasing temperature up to 4 degree higher, from 1.8 to 4 degree centigrade. So if this kind of 4 degree temperature increase, definitely we have to face lot of consequences in the environment. So the climate change treaty warned the world to at least indicate the temperature, to control the temperature to 2 degree, so that we can avoid serious danger consequences. So for that, what is the possibility of controlling the temperature at least 2 degree centigrade? The only way is biomass based energy. As you know that the fossil fuel also a biomass based energy, but the biomass buried inside the soil in deep several million years ago are converted into a fuel. Those fuel now we are taking out and we are refining and using. But now when we burn those crude oil, so automatically the carbon dioxide level is keep increasing. But to control the carbon dioxide increment, we have to only way to control it is by growing plant. When we grow the plant during the photosynthesis absorb the carbon dioxide, convert the carbon dioxide into carbohydrate. So that is biomass. During the photosynthesis, that is converting the carbohydrate into sugar, carbon dioxide into sugar, by it release oxygen. Okay, so this is the basic significance of the plant, why we bother or why we require as of now biomass based energy. When the biomass is used, the amount of carbon dioxide consumed for carbohydrate production is equal when we burn the biomass based energy for energy production. So there is amount of carbon dioxide consumed by the plant and amount of carbon dioxide when burn the biomass, it is released or nil. So the buildup of the CO2 in the atmosphere is zero. So biomass based energy is as of now required to mitigate the global warming. That is why we very much interested or very much require or we are demanding biomass based energy. If you see the plant biomass in general, through the three major type of conversion, we can produce any kind of energy which is we are actually getting from the fossil resources. Thermochemical conversion, by which combustion we can get heat, pyrolysis we can get bio-oil, coal and gas, gasification we can get burner gas through thermochemical conversion of biomass, plant biomass. Biological conversion, by fermentation we can get alcohol, ethanol. Anaerobic conversion, we can get gas, various gases, methane, ethane, butane, whatever gases we can get. Physical conversion, we can get oil. Using the biomass, we can get the biodiesel. This is the principle of plant-based biomass, energy conversion. But when we grow a plant, and if we try to get the energy from the plant biomass, there is an important challenge we are facing now. What is the important challenge? As you are engineering people, may aware, not much aware about the internal structure of the plant. The plants are erect as a big, long, big girth of the trunk, wood. Some plants may be having several meters of height. That is because of the strength. They do not have any bone as like animals, but they have the cell wall, lignin, the cellulose and hemicellulose, which are the sugar molecule present in the cell wall of the plant, which is given support by lignin. This lignin attached with, actually embedded in the cellulose and hemicellulose, which will not permit to convert into simple sugars. This cellulose is made up of polymer of glucose. Hemicellulose also made up of polymer of glucose. But this lignin is just sandwiched in the cellulose and hemicellulose in the cell wall. You can see various plants, grass, switchgrass, miscanthus, hardwood, oak, pine, spruce, corn stover, rice husk, carbon prawn. So these are all various plant-based biomass that can be used for energy production. But the challenge is that you can see the lignin content. This lignin content will interfere in the conversion process. But algae. So algae, I tell to you little more descriptive that based on the structure, algae are two types, microalgae and macroalgae. Actually, microalgae are very minute. You can see through microscope. By the mat, you can see by naked eye. But individual algae, you can see by microscope. Whereas macroalgae, if you are happen to go to the ocean, where you can see a plenty of algae which submerged as a forest in the marine water. So they are macroalgae. These microalgae and macroalgae are enormously growing, and they can support the biomass production, since they are like converting energy from the solar energy through photosynthesis and converting into biomass. But by what is another significance of the biomass feedstock compared to plant feedstock? As we know that we are very poor country, but highly populated, less space we have. So for already we are having a demand of land for cultivation of our food crop. When we need a large expand of land for cultivating a crop for fuel production, then it is very difficult. But algae can be grown in very limited space, but we can harvest large number of biomass. That is advantage. This is a algae as a potential biofuel factory where this light energy is converted into various energy like starch, ethanol, biodiesel through lipid production. Carbon dioxide is converted and oxygen is released. Even gas, hydrogen gas can be produced from these algal sulfides. So actually, this is my a comprehensive publication in Journal of Cleaner Production in the year 2019, where I have comprehensively given about the importance of the algal biomass for energy production. If you here you can see algae are, you may be knowing or whether algae are commercially nowadays used for pigment production, polysaccharides, polyphenols, animal feeds, so many things. As of now, so many cosmetics and nutraceutical compounds are produced from the both micro and macro algae. And the importance of these micro and macro algae, they can grow in wastewater and they can absorb the carbon dioxide and also absorb the heavy metals. So it is a good environmental remediating candidate. And as I told, these algae are the potential for production of biodiesel, bioethanol, butanol, methanol, hydrogen gas, whatever it is. It has potential. How it is practically possible, we will discuss. As I told, as like other plant biomass, algal biomass also capable of converting into various energy sources through biochemical conversion, thermochemical conversion, direct combustion or chemical reactions. And as of now, algae are used in cosmetic production, nutraceutical production, food, fertilizers, animal feed. But this feedstock can also be found potential for the production of biofuel. That we will see in details. These are all the microalgae. I told algae are categorized into microalgae and macroalgae. These are all the microalgae which are commercially used for the production of various costly biomedical compounds. Anabina, Menophilis, Chlamydomonas species, Dunaliella salina, Dunaliella piculinata, Euclina, Porphyridium, Senidesmus, etc. If you see the biochemical structure, biochemical composition of the some of the important commercially useful algae, you can see the lipid content is 21% in Chlamydomonas. Chlorella 14 to 24%. It is very high than the higher plants. Arthrospira, you can see the protein content 60%. And you can see here carbohydrate content is 13 to 16%. Synechococcus, cyanobacteria, very lower algae, you can see the protein content 63%, carbohydrate 15%, lipid 11%. This is very important commercially useful algae. You can see production of this beta-carotene. These are all the important compound, human consumption cosmetic compounds. The cost value of the beta-carotene from the Dunaliella cost is 115 to 2,150 euro per kg. It's much very costly. You can see Hematococcus pluvialis, another microalgae. It is commercially nowadays, it is grown in industries and they are extracting astaxanthin. It has a lot of biomedical value. So the cost is 7,150 rupees, 50 euro per kg. What is the actually, as of now, the algae are commercially used for various biomedical compound production. How this algae can be used as a source of biomass production, we can discuss. So I will take you in this aspect, because already these algae are growing. A well established growth methods are available for each algae, and they are there. After the growth, biomass is extracted. That biomass is used for the various biomedical compound production. This biomedical compound, after removing this, the biomass is residue is considered as a spent. It has protein, so that biomass having oil, and how this oil can be used as a source for bioenergy. What is the advantage of algal biomass than the plant biomass? As I told earlier, if you consider the biomass of the plant, first generation, second generation, third generation biomass is algal biomass. Actually, first generation biomass are sugarcane crops, potato. So they are food crop, but because of the sugar content, they were used for the production conversion of the ethanol. But when we depend this food crop such as sugarcane and potato for corn for the production of the ethanol, there is a shortage of food, and the price of the sugar will also get increased. To avoid that, the researchers found the second generation biomass, that is corn stove or grasses, especially switchgrass, some energy based grasses, which are not food crop. But those crop, when we go for growing in the land, highly populated small country like India and other developing countries, poor countries, where they need lot of land for food cultivation, there is a problem. So we need biomass, but that should not be compromised by the land which is used for the cultivation of food crop. So what is the alternate crop for biomass production? So it is, I say, or now it was realized algae is the suitable uh, third generation biomass crop. These algae are not a food crop, but they are as a source of various biomedical compound production. So food crop won't be get disturbed, and definitely we can grow the algae either in the ocean or in the freshwater, very limited space. Even we can grow algae in wastewater, not in freshwater. Whereas in the plant biomass, we need a freshwater to grow. It is non-food biomass. There is no lignin content in the biomass, as I told earlier. How the lignin is interfere the conversion of uh, biomass into simple sugar. Here, lignin is absent in the cell wall of the algae. So it is more advantageous for conversion into biofuel, that is hydrolyzation. And there is a data where you can see for when you go for soya or jatropha oil farm, the amount of land required is only less than 4% for the algal cultivation. in which we can get the biomass of the soya by using 300% land. But less than 4% land, we can grow the algae in which we can extract the biomass of the soya, jatropha, or oil palm. Oil palm required 23%, whereas jatropha required 75%, whereas less than 3% we can achieve. Because when we go for biomass based biofuel, there is 150 million total estimated biomass is required to meet the demand of fuel from the biomass. So where we go for the land? Very difficult. So algae can support. If you see the oil content in the various crop, the microalgae oil content is liter per hectare. That means 58,700 liter per hectare we can achieve the algae from the microalgae compared to other crop, whatever it is, first generation or second generation food crops. This I will tell later. But is microalgae cultivation is commercially feasible? Commercial large-scale production of algae biomass is not feasible as it plays on unsustainable demand on energy, water, and nutrients. However, obtaining high value because obtaining high value compounds and developing higher biomass producing strains strengthen the hope on the algal bioenergy production. As of now, actually, if you grow the algae in a particular wastewater and you go for the biomass conversion into ethanol, whatever is the biodiesel, economically not feasible as of now. Then what is the alternate? But as I told, no, when we go for the commercial pigment production first from the microalgae, then the residue can be used for the energy production. So the economical feasibility is very much achieved. If you grow the actually jatropha earlier some 10 years back, everybody was talking about jatropha cultivation for energy production. But now even the second generation energy crop like grasses, the economical possibility viability is limited because we have to we have to get a big land where we have to grow the algae plant and a lot of manpower and water, everything is economy is high. Whereas algal cultivation, when we get the biomass alone, economy is not that much. But if you go for more than one product development from the biomass, especially in algae, as I told, no, 1,200 euro per kg of particular astaxanthin pigment from the Haematococcus, Dunaliella, beta carotene from Dunaliella algae. So when we grow this algae and first if we extract the pigment, which is commercially important and valuable nutraceuticals, the residue can be used for the energy production. In that condition, there is a immense hope for the uh, biomass, algal biomass for energy production. And as this algal biomass, not as I told, it is not only growing in the fresh water but also growing in the wastewater. We can see here. So this algal biomass can also be grown in this is the microalgae cultivation in small simple open pond we can grow and we can extract the This is the spirulina cultivation. This spirulina is a good source for the protein content and phycobilly protein content. So this spirulina can be grown and we can extract the pigment and we can extract the protein. Then the carbohydrate can be subjected to ethanol production or any gas production, depends on whether it is subjected to thermochemical conversion or biochemical conversion. So actually this microalgal mass cultivation is developed in a photobioreactor in the open land where we can achieve economically viable biomass yield. But compared to this, it is a very cheap, less technique involved, and anybody can do cultivation. Whereas here it involves cost and technology, but we can achieve more biomass. And there are other method of cultivating the algal biomass like ocean, open raceway pond. Okay, so many methods are available. So open pond, we can grow the algae, we can cultivate. But as I told, now the researchers are aiming of transgenic plant, transgenic algae, or say through manipulating the gene. Because as I told, no, the algal biomass, the cost involvement is very high. The product which we are getting is very less than the cost involved for the cultivation. That is why the present algal engineers are suggesting more than one crop, more than one product development from the biomass of the algae. But in the sustainable way of energy production from the biomass of the algae, it is we have to develop a simple method of energy conversion from the biomass. So in that case, there is a much hope on gene manipulation of developing the biomass for the production of high oil content and high value pigment production from the manipulating the metabolic pathway. Here, as I told, this is the metabolic pathway where carbon dioxide enter in the cycle, Calvin cycle. It is Calvin cycle, and it is finally converted into fatty acids through acetyl CoA intermediate, where this acetyl CoA is transferred to the fatty acid production. Okay, so when we promote the synthetic biology of converting the uh, carbon dioxide into sugar and directly over expressing the gene, that is called over expressing, that is called synthetic biology, then there is a hope of increasing the production of fatty acid, where we can achieve. So this is the which actually the synthetic biology, they are aiming of promoting the particular gene over expression. Ultimately it will produce more particular product, for example fatty acid. This is the way, isolating the algae, selecting, this is the way of technology to find out the particular gene expression.
Specifically with the size.
These are all the algae they have attempted by various molecular technique of gene manipulating in the algae for the overproduction of the oil products from various algae. And actually, out of the various research in the world, in 2017, 2017, they achieved the by gene editing technology from the Nanochloropsis cygansia. It is a microalgae, Nanochloropsis. It is a marine microalgae. So this marine microalgae can grow in the open raceway pond by Nanochloropsis cygansia. For example, 20% actually the strain, this microalgae, don't know, the screen is appearing. This 20% actually, the actual strain give 20% of the oil production in the biomass, whereas by gene manipulation technology, it was achieved of 40 to 50% increase in the oil. So what that American company, they actually started cultivating. They are aiming of 11 million barrel a day in the year 2025, cultivating the Nanochloropsis cygansia by open raceway pond to achieve the oil production for the fuel demand. So they actually they set the goal of go for commercial production in 2020. But in practical, they actually first announced in 2020, but they postponed to 2025 because the strain they developed in the pilot level and the yield in the large scale raceway pond, the yield get decreased. Actually, since it is a biological system, we have to see lot of other environmental factors which involved when we grow the algae in the various climatic conditions. Actually, they are nearing of producing the biofuel by using this Nanochloropsis algae to achieve the demand. As of now, 10,000 algal strains have been collected, classified, and screened, and these algae found as a potential for various energy production as well as the bioproduct development. But we invested a lot of money and we are hoping for developing the photobioreactor or open raceway pond technology for economically viable method of producing the biomass. When actually that American company, they set the goal of producing the oil from the Nanochloropsis in the year 2025. When they demonstrated it, there is an immense scope of growing the algae by using the pond, open pond like this. So it is a simple way of cultivation. Then as you know that actually when we get a biomass of the tree, we have to grow the tree for nearly 20 to 30 years to get a big biomass. Whereas in microalgae, in a small pond, if they are keep growing and we can achieve within a year, yet 30 years biomass of the tree can be extracted from the pond, small pond, by cultivating microalgae. But it involves care, not like a tree. For that, the technology like raceway pond, photobioreactor, open pond, simple pond, we can grow by using the manpower. When we involve the manpower and the technology, it involves high cost. But this biomass not only be used for the energy production, but also we can obtain the bioproduct like pigments, nutraceuticals, etc. So then economically possible uh, method developed from this microalgae cultivation can be commercially practically propagated to common people to use this algal cultivation for energy production. And as I told, no, there is a after growing the algae, after obtaining the biomass, how can we extract the energy? How we can extract oil? How we can convert the bioethanol? So that is another research, another method. It is highly developed. I will tell you. So it is operational. Wait. Ah. See, actually, this is the method they are aiming for. This is the method they are aiming for converting the biomass into product in a simple way. So the conventional way of growing the biomass, harvesting, then cell rupturing, extraction, fractionation, product development. It is the conventional way. After getting the plant biomass, so from the algae, we have to harvest the biomass from the pond or raceway pond or whatever it is. Then we have to disrupt the cell, break the cell wall. We have to release the compound, either by mechanical method or chemical method, catalytic method, like that. Then after grinding, we have to extract the compound. Then the extraction by, it may be by solvent extraction or uh, chemical extraction, whatever it is. Then we have to separate into ethanol or oil, or as I told, no, various bioproduct. But see, this involve various steps. Then cost involvement is there. Now the method achieved is harvesting and cell rupturing together. It is achieved now. Then that means when the algae grown in a pond or open raceway pond or in the photobioreactor, after the harvesting stage, we can go for so many method, autoflocculation or cell rupturing inside the in situ level. Then from there we can extract go for that biome, all the cell get ruptured, then we can extract the product and separate it. But the further research technique is aimed of harvesting, cell rupturing, extraction, fractionation on the spot. When algal grown at maturity, that level itself, if you do all the technology, then we can get the product. So the cost involvement for post, that is downstream process, is less. So the possibility of algal biomass for, or feasibility of algal biomass for fuel production is more promising. So with this, I will go to the next topic: how this algae will support the various biofuel production. Various biofuel production. The new slide is visible.
Sir.
Actually, okay, thank you. So as I told, no, this biomass, algal biomass, actually I made some research. I have actually developed a pilot scale production of ethanol from the marine algae biomass by cost effective method of using the marine bacteria and marine yeast. So in this, actually, you may be or whether you know the information or not. This agar, you may be knowing agar, which is used for growing bacteria in the laboratory. And if you are a biology or biotechnology, bioengineering people, you may have aware. And the DNA separation and all, agarose is used. And you may be knowing the food items, jelly, jam. These are all prepared from the agar. So the source of agar is algae, marine algae. They are called marine red algae. So actually it has a huge demand. If you go to this marine red algae are collected from the ocean and it was given to the industry, they are extracting the agar. It is a very high value product. The residue are thrown into the environment. So I found the residue containing lot of sugar. That sugar residue are subject as like a bagasse. How the sugar cane given by the farmer into the sugar industry. Actually, the farmers are given the cane for the value of the sugar. But the sugar industry people get more money from the bagasse when they go for ethanol production than what actually they are getting as a sugar. That means the revenue for the industry is triple, whereas the farmers given their sugarcane crop cultivated by lot of difficulties to the industry by very cheaper cost, very low cost. You may be knowing that in Punjab and Haryana, the paddy and wheat waste are residues after the harvest left in the land. They were burned because of when they go for next crop rotation. But government, if they support to the farmers and government permit the farmers and support the farmers, there is a immense scope of ethanol production from the residues. But the government readily and really given this permission of converting the residue into ethanol, but there is a problem of ethanol production from the sugarcane bagasse residue, and there is a definite decrease in the ethanol cost also, and the farmers will not burn the residue of paddy and wheat, especially in the near Delhi northern district area. So in my research, I found that a residue of the red algal biomass, which is after extracting the agar, can be converted into ethanol by cheaper cost, very low cost. This we have seen it. Actually, what is the significance, the necessity of algal biomass of fuel production? When we go for the ethanol, actually, when we go for algal based biomass production. So that biomass, when we convert into various energy, if you go for ethanol, actually earlier told ethanol is suitable for transportation fuel, but ethanol now it is corrosive. So existing fuel specificity of the machine will not be conducive for ethanol blending. Okay, so then what is the alternate fuel? Is there alternate fuel available for the uh, ethanol or biodiesel or methanol, which is suitable to existing engine specificity? So those kind of fuel, is it possible to get from the algal biomass? It is not possible to get from the plant biomass. It is possible to get from the algal biomass. If you see the corrosivity and hygroscopic nature of the ethanol, existing fuel storage and distribution infrastructure in the engine is not suitable. Then the establishing, or if you go for designing a new engine system, it incur lot of cost. So high, that means importantly its high octane content in the ethanol. Only 70% of energy content is present in the gasoline. Okay, that means energy content also less. So how we can go for the good energy content energy, which is low corrosive and low hygroscopic in nature? From the algae it is possible. So that means advanced biofuel having high energy content and less octane number, we can go for production of this kind of future algae, future oil from the algae, compatible with the storage and transportation infrastructure designed for petroleum based product. It is also economically feasible to produce on industrially large scale by using algae. That means a similar property of petroleum based, gasoline based fuel, but it can that will replace the petroleum. So such kind of fuel we have to identify and we have to produce. In that, for example, there are gasoline, diesel and jet fuel. These three can be replaced by a latest fuel or new fuel which is more than suitable like this fuel. So how we can do it? So by microalgae it is achievable, and a lot of research is going on, and there is a way we can achieve it. I will explain about that. So if you take the gasoline, is a mixture of C4 to C12 hydrocarbon, linear branched cyclic alkane composed of 40 to 60% of the fuel mixture. It contains aromatic compound of 20 to 40%. Biofuel so what is the suitable alternate to gasoline? It should contain 32 megajoule per liter. Transportability must be there. The octane number should be high, that is 87 to 91%. Then a potentially advanced biofuel capable of supplementing or replacing gasoline should be a short chain alcohol and alkanes. So what is the suitable gasoline alternative? Is a short chain alcohol or alkane. It can be synthesized from this dunalial. As I told, this dunalial already used as a source of commercially important nutraceutical compound. So 1 kg of the pure compound, it depends on the purity. The value of the compound is high. Even 7,150 euro per kg of this beta carotene uh, from the Dunaliella, which is a marine. From this microalgae, there is a possibility of accumulation of C15 to C19 alkane, which is suitable alternate to gasoline. So the yield of the alkane as of now is 0.2 to 1.12% alkane gram per gram cell dry weight is achieved. So you can see how there are cyanobacteria which are also supporting of this short chain alkane synthesis. If you come to the biodiesel, so the diesel is having the 9 C9 to C23 hydrocarbon, which average chain carbon length of 16. It is a linear branched cyclic alkane composed of 70% of the fuel mixture. The aromatic composes of 25%. What would be the suitable biofuel for biodiesel? That is, the quality of the suitable alternate biofuel for biodiesel must contain freezing temperature minus 9.5 degrees Celsius. Vapor pressure should be 0.009 psi at 21 degrees Celsius. Cetane number must be 50 to 60%. So this quality can be obtained from the fatty acid methyl esters and fatty alcohol, alkane, and linear and cyclic isoprenoids. This can be synthesized, or these are all synthesized from the microalgae. So these microalgae hold potential for large-scale production of biodiesel because of its non-competing nature with food security, as I told, and it yields more biodiesel per hectare of land utilized. Growth rate and lipid content of the algae is very high, as I told. Compared to the other crop, the algae require very less land. Algae used for biodiesel production are mostly belongs to the group of Bacillariophyceae, Chlorophyceae, and Chrysophyceae members. So actually, most commonly used catalyst for the fatty acid methyl ester development, a cheaper catalyst like sodium hydroxide, methanol, and potassium hydroxide. If you see the jet fuel, the jet fuel gas turbine is a mixture of C8 to C16 hydrocarbon. Jet fuel has a maximum of 25% aromatic compounds. So what would be the suitable jet fuel alternate which is available, or which can be synthesized from the microalgae, or fatty acid and isoprenoid based biofuel from bacteria and microalgae? So this by engineering of microalgae, there is a immense scope of producing such a advanced alternate fuel which can replace the gasoline, biodiesel, and jet fuel. They are alcohol, esters, alkanes, and alkanes from isoprenoids and fatty acids. So this can be possible by manipulating the metabolic pathway. If you see the Synechococcus, and this is the cyanobacteria where actually this acetyl CoA. So how, for example, how in the circuit it is a you imagine this is a biological circuit where, yeah, in a transistor in a place in a like that, this is the in a circuit, but here it is a metabolic circuit where the product is alkane formed through intermediates various intermediates. So when this fatty acetyl CoA, it is triggered to over synthesize, subsequently it will release this compound alkane. And we can also improve the chain length. The same head to head condensation, if you go, the chain length of the hydrocarbon is C23 to C33. Here we can improve the chain length again by C24. So how we are triggering the metabolite, how we are uh, stopping one metabolic circuit, and we are diverting the acetyl CoA to here. The sugar is used for growing the algae. This sugar can be a waste sources. This cyanobacteria can grow by using a waste water or waste land, by using only water. So it will convert the solar energy into sugar, or waste sugar can also be used, and it depends on our requirement. We can stop this metabolic circuit, and we can permit to uh, produce only this alkane. Suppose if we want this alkane compound, then we can scissor this metabolic circuit. So this can be possible by using a microalgae. So when we achieve this kind of gene manipulation technique, then we can sustain of producing the our advanced required fuel by using the biomass or organisms. So industrial fermentation can readily be applied by using microalgal production of advanced biofuel in another category, where we can produce the microalgae biomass. That biomass we can subject into fermentation, where we can produce various gas. So these are all the yeast, other bacterial biomass, E. coli. These are also can be or not can be. It is genetically modified, and these organisms are cultivated for the production of the specific hydrocarbon. Here you can see this is a one kind of a metabolic pathway exploited for the production of known and potentially advanced biofuel. As I told, this ethanol from the acetyl CoA, we can trigger the alkane, fatty acid esters, or 3-methyl pentanol, butanol. So whatever the fuel required, that can be possibly produced by triggering the metabolic circuit of the microorganisms, especially cyanobacteria. So this is another gene expression technique by which E. coli and Saccharomyces yeast and another bacteria, Clostridium, are manipulated to produce isopropanol and 1-butanol from acetyl CoA. So actually, this year in the beginning, we published this paper on seaweed. As I told, no, the marine algae are called seaweeds. The biomass of the marine algae after extracting the uh, agar and alginate. There are different marine algae. So this biomass, gracilaria biomass, used for bioethanol production and followed by yeast by very cheaper biosaccharification method. So the Clostridium bacteria is used for isopropanol production by triggering the uh, metabolic circuit or metabolic pathway. So isopropanol is a C3 alcohol, has an energy density of 23.9 megajoule per liter, somewhat less than that of gasoline, a high octane number, and low water solubility when compared to ethanol. So isopropanol, which is suitable than the ethanol, is possible of produced from the isopropanol. So for example, ethanol, actually when we get the ethanol, chemically from the ethanol we can produce so many other compounds like butanol, algane, isopropanol, propanol. But it chemical conversion by catalyst, it again what happen na, it will create pollution. It involve lot of chemicals. Whereas naturally, if organisms are manipulated genetically and the organisms are capable of producing the our required various alcohol, then this can be easy and eco environmental friendly, and it mitigate the global warming, and that means CO2 buildup in the atmosphere can be controlled by growing this algae, and we can achieve the energy production. Isopropanol is currently based on gasoline and biodiesel additive, but Clostridia isopropanol pathway require 1 mole of glucose to produce 2 mole of uh, isopropanol. So that means it is good depending on the Clostridium for isopropanol production. Then butanol, it is another energy, advanced energy fuel. C4 alcohol is a biosynthetic alternate to gasoline. Butanol has an energy density of 29.2 megajoule per liter, which is comparable to that of gasoline. Octane number is 87 and can be mixed with gasoline at any percentage or even completely replaced. Gasoline can be completely replaced. This butanol high hydrophobicity may allow the use of existing fuel transportation and storage infrastructure without major modification. So Clostridium butanol pathway require 1 mole of glucose and 4 mole of NADH to produce 1 mole of butanol. So the Clostridium pizargici is capable of producing 19.6 gram per liter of butanol. And there is another potential biofuel production is identified by rerouting the amino acid pathway. As I told, no, this metabolic circuit can be either diverted or rerouted, or any metabolic intermediate can be over expressed or over permitted to overproduce for the particular product development by various gene manipulation technique like gene editing, overexpression, or triggering, etc. Another way of rerouting amino acid biosynthetic pathway for production of higher alcohol by yeast. Here we can also use yeast for the production of N-propanol, N-butanol, 2-methyl 1-butanol, or 3-methyl 1-pentanol by manipulating the amino acid pathway. So this turbine compound can also be produced, which is suitable for jet fuel replacement, including pinene, saponin, and turbinine. So these are all possible by producing metabolic engineering of isoprenoid biosynthetic pathway for the generation of this advanced jet fuel. So the suitable advanced jet fuel can replace the jet fuel or pinene, saponine, and turbinine. So this advanced fuel can be produced by using the cyanobacteria and E. coli. So this MEV and DHP pathway for the production of isoprenoid. So this fatty acid and pyruvate pathway are the potential pathway. It can used for the production of jet fuel like isopentanol, turbinine, sesquiterpene, so monoturbine, so titurbine. So this can be possibly produced by rerouting the fatty acid or pyruvate pathway in the cyanobacteria. So this metabolic engineering of fatty acid biosynthetic pathway for the generation of fatty acid based biofuel also possible by using cyanobacteria. So that means how this fatty acid can be converted into biodiesel via esterification with a small alcohol. So this is the way how we can reroute and we can elongate the fatty acid chain also. Depends on our requirement. We can, for example, carbon fatty acid chain. See whether it is saturated or unsaturated, whether the chain length is C15 or C16, whatever it is, according to our requirement, we can manipulate and we can produce. So these are all the way, these are all the microalgae identified, and possibly we can produce various fatty acids in different chain length. Here you can see linseed oil, soybean, fish, hair plants, this. But this Zenidesmus, chlorella, nanochloropsis, where we can produce all these fatty acids. Very high number than any other fat, any other plant sources. Here you can see the composition of the sterol isoprenoid derived from hydrocarbon in unsaponified lipid of 3 algae genera. So this, as I told now, this chlorella, Zenidesmus, and nanochloropsis. You can keep in mind these 3 algae are very much commercially promising biofuel producing microalgae. From this, lot of gene manipulation research is going on by using this algae. As I told, the commercial biodiesel production in US is going on by using this nanochloropsis agentia. Here you can see the various amino acids production from various strains of this Nanochloropsis, Chlorella vulgaris, Cyanidesmus, where this has lot of potential for diverting the amino acid pathway, as I told you earlier, for the various this octane alkane productions, which is considered suitable for the jet fuel production. So I hope that I could address some information about the potential of the microalgae for the future biofuel demand. I can say that why we in a concluding stage, among the biomass, this microalgae biomass is a promising third generation crop. This biomass, this microalgae or this algae, not only microalgae, macroalgae, it is not directly used as a food for the people. So cultivating this algae for energy will not affect the food crop. And this algae cultivation, either microalgae or macroalgae, we can fetch income and economic feasibility is possible if we go for more than one product development. Actually, now our era, we are aiming selective food product consumption. For example, protein consumption or nutraceutical consumption or some selective bioproduct for selective health benefit. We are taking multivitamins or bioproduct. So in that, this microalgae cultivation will support a selective nutraceutical demand for the people, as well as the biomass generated can be used for energy conversion. And another point depends on our demand of the energy, specific demand of the energy. The microalgae can be manipulated through gene manipulation or genetic engineering technique, and we can produce the particular fuel by using the microalgae. And unlike the higher plants, this algae did not require vast land for the cultivation. It required only very limited space. And I used to tell that cultivating the tree or grasses energy crop, it is little cumbersome. It require fresh water, bigger land. But cultivating algae is a kind of techniques involved, simple, but there are various method of simple open pond cultivation, raceway pond cultivation, or photobioreactor, whichever you are selecting. And accordingly, you can go for the a specific product development. When we want to a product which is used for consumption, then you have to aim either raceway pond or photobioreactor. So we can cultivate the algae in a very aseptic, free of contamination environment. So the product can be used for consumption. So as I told that this microalgae are a promising crop for future sustainable energy, not only energy production, for other nutraceutical product development. Definitely a cheaper technology development and more uh, product yield achieved by using the specific strain. When we achieve, then microalgae is the ideal and promising candidate for our future energy demand. With this, I request, if you have any doubt and clarification or question, you can ask. We can have a discussion.
So dear participants, please unmute yourself and ask any questions, any doubts in this presentation. So sir is waiting for the questions.
And actually, I can tell that my MOOC course is available
Yes,
in
sir.
Swayam platform. Actually, it is describing about all biomass and all energy. I have gone through the title of the talk in this program. So my MOOC course will cover all the topic. So if you now the course is available for the enrollment. Biomass, the course name is biomass characterization, and you can go through the videos, and the content is available, and all method of algae cultivation, all these various biomass energy production, various energy conversion process, source of energy biomass, what are the method of characterization of the biomass. I think I can say it will cover your topic which you need if you want to have an idea about the biomass source for energy production. I request the participants to enroll and you can learn the biomass characterization MOOC course, which is available in the Swayam platform as of now. Thank you very much.
Participants, any questions?
You can ask
Participants, please,
freely. You can
please
ask.
unmute yourself.
I understand the difficulty of the participants sitting nearly 2 hours, and it is also difficult for our side to present continuously for
Yes, sir, for 2 hours it is very difficult.
without seeing the face of the participants.
Yeah. It is a continuous deliberation is very difficult. No dialogues, na?
Yes, I request the participants to go through the MOOC course. You will get more idea also. You freely ask any questions. I ask 1 or 2. I expect a few questions from the participants. No, I don't want a nice presentation. Some are putting.
Hello, sir.
Yes.
Sir, how to clean this algae when we extract from the source, and how to use it for the production of biodiesel, sir?
Actually, when we grow the algae, as I told, no, if we plan of producing biodiesel and any other consumable product, then we have to find out a suitable uncontaminated environment for the cultivation. For oil production, there are a lot of methods available. Simply, the biomass should be ruptured by mechanical grinding, or it can be subjected to, for example, microwave disruption, or catalyst crushing, or so many other methods are physical method, crushing the biomass, then oil will release it out. This oil we have to convert into fatty acid, methyl esters, for the biodiesel usage. Oil as such we cannot use it. We have to convert into methyl esters by using NaOH or KOH or methanol. So that is a esterification process. But extracting algae from the algal extracting oil from the algae, you can use chemical method, physical method, or biological method. By cell rupturing, even enzymatic method. Cellulase can be used, added, and the cell wall get ruptured. The oil present in the lysosome of the cell will come out. Are you satisfied?
Yes,
Are you
sir.
convinced?
Yes, sir. Thank you, sir.
Still any questions from the participants?
Yes, I have a question.
Yeah, please.
What are the methods for this growing this algae?
Actually, as I micro, for example, in macro algae, if you which is your native place?
I am from Kerala itself.
Which location?
Kottayam.
Oh, Kottayam. Actually, in I am trying of cultivating marine algae in the ocean. Cultivating marine algae in the ocean, just you can put a rope. If you happen to go to Rameswaram, where you can see cultivating algae by net and throwing the net in the ocean, by inserting the algae thallus, it will grow. So macro algae cultivation. And the micro algae cultivation is by, as I told, even your home, by a cement pond, small pond, and your utensil washing water alone sufficient for leave it as such, algae will grow. If you are introducing your choice of algae, any wastewater or wastewater with soap, it is some medium is available. Depends on the choice. For example, if you are planning of cultivating algae, and there are nutrients are available, inorganic nutrients alone you have to add. Calcium, potassium, sodium, some salt little you have to add. By using the solar energy, it will convert and it will grow. So biomass, we have to periodically harvest the biomass. Harvesting techniques, so many techniques are available for harvesting the biomass. But that is a simple open pond. Whereas photobioreactor, large glass tubes are available, where algal, small amount quantity of algae you have to introduce with full of water. So this glass tube will get penetrated the light, so algae will grow by using the water available. And by small filter opening, we can withdraw the water and we can harvest the algae, then biomass. Then again you have the same water you can recycle into the photobioreactor. There is a system is there, that is highly advanced photobioreactor, where you can cultivate the algae. And pond, that is called raceway pond. I think you might have seen the prawn cultivation. Prawn cultivation is a less technically involved pond with aerator, pedaling and all. But this algal cultivation pond is a cement pond, where water will keep circulating by motor, so algae keep growing. After the reaching the growth, there is a technique of sedimenting the algae, by that is called autoflocculation. Then automatically we can harvest the algae manually, or by siphoning through the motor. Then again we can permit the algae to grow by using the or adding some medium, further medium, because same water, if you are repeatedly growing, the medium water will not have sufficient salt is required, some nutrients. Salt means nutrients, that we have to provide very little quantity only. So algae can be grown by small pond, raceway pond, photobioreactor.
Okay, thank you, sir.
If you happen to go to Rameswaram area, where you can see lot of marine algae cultivation. There people are cultivating and they are supplying to the company for this high value product development. The remaining biomass they are using for ethanol conversion or some compost product development. I actually next year I am planning of doing the algae cultivation in Villingam area in Thiruvananthapuram. The project is we are doing, it is underway. Because of the corona it is postponed.
Okay, thank you, sir.
Actually I was collaborated with environmental research laboratory of Adani Group in the Villingam, where the fishermen's life support can be helped by growing algae. That kind of project I have taken up with one scientist in that Adani Research Environmental Research Center at Villingam. I think next year people can see who are coming to Villingam.
Still, anybody want to ask questions?
Sir, nice presentation, sir. Is there any side effects by using algae? Is there any side effects?
Side effect for what? Consumption or?
Consumption also in case of any kind of any health issues in case if you are contact with that particular.
Actually, for example, I told no beta carotene, astaxanthin. If you take there is one pigment you note down astaxanthin. It is produced from the or extracted from Haematococcus microalgae. It is a very high antioxidant compound. It was not reported. None of the algal compound, commercially useful compound, not toxic. It is used for human consumption. People are using this astaxanthin is very suitable anti-aging compound, and it is a natural colorant. It has high antioxidant potential like vitamin C. Okay, then beta carotene, which is extracted from Dunaliella, another microalgae. It is a natural colorant. It is used for various food product colorant. Okay, it is very costly. It is not toxic. And phycobilly pigment, which is used as a natural colorant for various food product. It is also not toxic. It is available. People are using, and very costly, and high food product are colored or given color by using this compound. There is no toxicity reported, because when we take the plant as a food, we are consuming some cyanobacteria. So no doubt these algae are not toxic. And marine algae are used as a food, no toxicity. But we have no capita of taking the marine algae as a food, salad as such. But in other country, all these European countries, Americans, Asians, not us, Malaysia, Japan, China, they are consuming the marine algae as a food. But we are consuming only agar, their product we are taking. But the other country people are taking algae as such, as like a vegetable, as we are consuming green vegetables. The other country people are consuming algae as a vegetable, salad.
Any questions?
Sir, in some of the, I can say drainage, okay, you can say pond, flowing water, stored water, okay, we can see naturally, automatically algae will grow. Okay, so like which one is more, I can say kind of nutrition, okay, which one we recommend to go with the natural, I can say, biogas production, biodiesel production.
Yes, yes. That's why I told you. Very good, very good. Actually, if you go to the natural place where the rain is a stream or stagnant water, you can see plenty of algae. Okay, but that algae we cannot harvest and we can't eat. Or we have to selectively grow the algae. When, for example, Chamatococcus, as I told, astaxanthin, beta carotene, Dunaliella you have to grow. Then spirulina or Synechococcus, if you are growing, you will get phycobilly pigment. Or chlorella, so you will get a lot of protein compound. Or chlamydomonas, if you are growing, you will get oil. Nanochlorapsis, you can get oil. So when you want to have, or when you go for a particular product development, according to your choice of the product, you have to grow the particular algae. For growing the particular algae, for example, you want biodiesel or oil production, you go for nanochlorapsis cultivation. Nanochlorapsis cultivation, yeah, algae is available in various research institute or universities. Even if you put it in Google, you can see the source of algae. You can purchase from Amazon also, available. You can purchase all the algal product from Amazon, beta carotene, astaxanthin, whatever it is. So then you have to go. The techniques are described. Even where you are purchasing algae, they will tell the method of cultivation. Then you can cultivate. But you need a knowledge for cultivating the algae, and you need high cost. It is not like a paddy cultivating the open land. So the technology now available is in high end. It is not in the cheaper open level as of now not available. So it will come in the near future.
Thank you, sir. Thank you so much.
Still any questions? Sir, I think no questions from the participants.
So, do you have any idea about ancient history of algae? Okay, because nowadays we come to know. Okay, still some of the people those who are not into in this field. Okay, they think this algae is no use. It is only irritation. Okay, when we go to like a pond and all. Okay, we see it is slippery. Okay, we cannot stand here by that. Okay, we have such kind of mindset. Okay, so what is the ancient history of this? Okay, how it has started? Okay, who has found this? Okay, like if you
Oh,
share, if you share that.
actually, very nice question. I am really happy of asking this question. Actually, I tell you, you people are engineers, no?
Yes, sir, that's what.
Algae is the our primitive plant. Plant we are seeing this angiosperm plant, that is trees, paddy, and all. Okay, this evolutionarily from the algae only these higher plants are evolved. As we say that humans are evolved animals, then other animals, the same way algae are the very primitive first formed plant. From this plant only, from this algae only, the other plants are formed. Okay, first. And since it has long history of using algae as a food, it is not in India. But now only science improved and we are realizing the importance of the algae. Algae not only used as a food, but algae support agriculture. When we go for growing rice and paddy in the field, you can see a algae mat inside the paddy land water. So this algae fix the it is a good source of nutrient for the paddy growth, crop growth. Okay, and in the soil, as you said, no, the slippery and all. Actually, it produces the
Yes, sir.
compound polysaccharide. Okay, so mucilaginous compound. That compound only it show the slippery in nature, but they are all economically useful, and the compound are very biomedically important. And if the algae are not growing, that soil will get affected. So it promote the soil fertility, and it algae grow, other organisms grow, and it is good for the plant growth when algae is growing in that soil. So ecologically, algae are very much supporting the growth of the other organism in the particular soil. Crop also, other crop growth also algae is promoting. And very importantly, in pond and ocean, all the fish are fed by algae, and the fish nutrients are algae. So if we are interested of eating fish, fish is interesting of eating the algae. So algae are the feed for fish. So without the algae,
Okay, this is one of the major.
yes.
Okay, this is one of the major part. It takes major part in biological cycle.
Yes, yes. Ecological. So algae playing a very crucial role in ecological cycle in all the level. Now we are considering its immense growth potential. We are exploiting. Now science is improved, and we are using this algae for our direct benefit by as a food or as a energy. How, for example, now we are consuming rice and wheat, but this took several million years for cultivating the rice in the land by various technology. So now we are using the rice as a food, paddy as a food. But before that, people consumed various other wild eatables from the plant. The same way, now science is improved. We realize the importance of the algae, and we are extracting some product like pigment and chemicals and biomedical compound for pharmaceutical value, medical value, and food value. Beside, we are aiming for energy production from the microalgae and macroalgae. Are you satisfied? Are you
completely
convinced by-
satisfied and yes, sir, completely satisfied. Yes, thank you.
Okay.
Still anybody? Still anybody?
I am happy of interacting with the participants.
But Chetu is an active person in all the sessions, sir.
Very good. Very good.
Okay, sir. No questions from the participants. So,
I thank you very much. Thank you very much for giving the opportunity of sharing my knowledge with your participants.
thank you, sir. First of all, we would like to thank you for accepting our invitation to give a lecture today. So you have given a lot of information to the participants, how to produce the biodiesel from the algae, biofuel. So thank you, sir. Once again, we'll meet you in the future.
Thank you very much. Thank you.
Sir, I sent one email to you. You please go through that one, sir.
Okay. Thank you.
Right, sir. Thank you, sir. Once again.
Thank you very much. Thank you.
So you can leave, sir.
Bye. Bye.
So dear participants, so this session ended today. So tomorrow we'll have the second week starts. Second week first session by Murli Mohan, and there is a virtual visit of solar dehydration plant established in an incubation center at RGMs at Nandyal, and in the on renewable energy technologies, fuel cells and flow batteries. So all of you join at 10:30 a.m. tomorrow. Thank you.